chills, fever, sneezing. Everyone is very familiar with these ailments. They are all symptoms of the flu. But what causes the flu? What is the flu? In this video, we'll tell you. We'll describe virus replication and what a virus is.
A virus is a non-living collection of nucleic acids and proteins that uses living hosts in order to survive and reproduce. It hijacks other cells, injecting its own genetic code and having the cell's organelles produce the components necessary for the virus's survival. For example, influenza, a common RNA virus, undergoes a specific process in which the virus replicates its RNA genome inside a host cell. There are three main steps during RNA viral replication: attachment, penetration, and replication. During the process of attachment, hemagglutinin that surround the viral cell binds to salicylic acid receptors in the host cell. Hemagglutinin have specific signals that notify the virus when a host cell is near and uses a lock and key method to stick to the host cell during replication. Once the virus attaches to the cell, it injects its genome or RNA genetic information into the host cell. The genome is surrounded by a protein capsule that makes sure all the genetic information is not lost during the transfer, then dissociates once the relocation is completed. In replication, the viral genome is unwound and an enzyme called polymerase goes through each single strand of RNA and adds complementary base pairs. By this process, the RNA injected from the virus is then replicated into the host cell. Protein capsules would then surround each set of the RNA genome and remove itself from the host cell. Thus, the cycle repeats and continues until an immunization of the virus is created.
You might be asking yourself how your body manages to protect itself from these nasty viruses. The body produces antibodies that recognize foreign invaders by looking for antigens. Antigens are proteins or indicators that are found on the surface of the pathogen. Each antibody has a unique binding site shape which locks onto the specific shape of the antigen. This means that if there is any type of change in the shape of the antigen, whether due to a frame shift, silent, nonsense, or missense mutation, the antibody would no longer be able to recognize and bind to the antigen. This is called antigenic drift. Small changes that occur in viruses over time produce new strains, and therefore the antibodies can no longer identify the antigens. This causes vaccines to lose effectiveness over time.
Flu vaccines are often mass-produced by injecting certain viral strains of infectious influenza into chicken eggs. Over time, the strain adapts to the new organism and loses its ability to infect humans. Humans are then inoculated with this new strain, allowing their immune system to learn to recognize the virus while suffering none of these symptoms of the disease. The immune system produces antibodies that recognize certain indicators on the surface of the virus called antigens. For influenza, the antigen that is recognized is usually the protein hemagglutinin located on the surface of the virus.
This brings us to a research article written by Skowronski et al. Concerning the effectiveness of influenza vaccinations from 2012 to 2013.
A sample population of 2,024 individuals in the 5 most populous provinces of Canada displaying flu-like symptoms were used in the study. Swabs of the patient's nose or throat were collected, and the patient was asked to provide information regarding their immunization status.
The flu vaccination studied in the research article, called the trivalent influenza vaccination, contained 3 major strains of the influenza virus to provide immunization to a wider variety of strains. These 3 major strains are shown in Table 2, along with the relative abundance of each strain in the population. Included in these 3 varieties is a strain called H3N2. Of the 3, it was found that the vaccine was least effective for the immunization of the H3N2 strain, even though labs throughout the area reported they detected no antigenic drift in the H3N2 influenza strain.
Since the H3N2 strain was not experiencing noticeable antigenic drift, scientists set out to investigate why the vaccination effectiveness was so low. They began by characterizing the virus samples. The viruses were then analyzed genotypically and phenotypically in relation to their antigens. The presence of the influenza virus was determined using a real-time reverse transcription polymerase chain reaction. The genotype of each virus was analyzed by direct amino acid sequencing of the HA1 protein, and the phenotype analysis was performed by the characterization of viral activity in gerbil and turkey cells, and also by using antibody titer tests. Figure 2 from the study shows a distribution of the strains of influenza that were detected in the population during a given week of the flu season. On the x-axis of the graph is the week, and on the y is the abundance of the strain. Each color represents a different strain, with H3N2 representing the most abundant strain. From the graph, it can be seen that the influenza activity peaks around week 2.
Using a test-negative case control design, the overall vaccine effectiveness was found to be 50%. Vaccine effectiveness for the H3N2 strain was found to be 41%. In the analysis of the genotypes and phenotypes of the various influenza viruses tested, numerous genotypic differences were found between the strains circulating throughout the population and the strains found in the vaccine. These tests indicated that there were distinct phenotypic differences in HA1, a critical membrane-bound protein of the virus.
Figure 3 compares the differences in the HA1 protein between the strain of influenza in the vaccine and the H3N2 strain of influenza circulating through the population. The HA1 protein, also called hemagglutinin, is a protein that our immune system can learn to recognize as an antigen, allowing it to attack the virus. However, as seen in the figure, there are distinct differences between the two HA1 proteins, leading to the inability of the antibodies generated from the egg passage virus to recognize the HA1 protein and the circulating H3.2 strain.
In the end, the study is important because it provides a possible explanation for decreased influenza vaccine effectiveness. If decreased effectiveness is actually being caused by antigen mutations during the vaccine production process in eggs, then alternate production methods can be pursued to increase the effectiveness of the vaccine and reduce influenza infection of the population. Furthermore, the study provides the opportunity to relate antigenic drift, genetics, viral and RNA replication, and other biological principles to the real world.